Welcome back. We are starting with chapter 14 and we will only consider here the section 14.1 through 14.3, regulation of bacterial cellular processes. So we have so far talked about how that the replication of the DNA, the transcription of the DNA into the RNA and the translation work. In this particular chapter, we are going to look at that how this process of transcription and then the translation is regulated. And there are many different types of regulatory options that we are going to cover a couple of them. So what are these regulatory options? These regulatory options can involve the transcription initiation, that how this transcription will occur or not, whether the transcription elongation will take place or not. That means that if the initiation of the transcription takes place, whether the mRNA will be produced in full length or not, that's another regulation. And then we can also regulate the process at the level of translation, because in the eukaryote, the two processes are not coupled. Whereas in the prokaryote, what we mean by coupled means in the prokaryotes, the transcription and the translation can occur simultaneously. We have seen those pictures of the polyribosome that as the transcription or the transcript of mRNA is being made, automatically they are being engaged by the ribosomes and simultaneously the transcription and translation can occur. So we can actually control at the level of translation. In the eukaryote, the two processes are separate because you have the nucleus where the mRNAs are formed. Those mRNAs need to come out into the cytoplasm and go into the ribosome. Entirely different spatial arrangement. So the transcription and translation are two separate processes, not coupled like in prokaryotes. So also there are some processes which we called as a post-translational modification as well. And this is for this particular chapter, we will omit this part. So again, there are three domains of life differ in the genome structure and regulatory mechanism used. Here we are particularly interested in the understanding of the bacterial regulation. We are not going to talk about the archaeal regulation. So here just gives you an overview of that, what is the process of the transcription and the translation. And if you actually look at that you can have this nucleoid associated protein and increase and decrease the transcription. There are some transcription regulatory proteins that can call, and then there's a process which we called as an attenuation of transcription that can happen. In the translational level, there are some secondary structure formation at the 3' end of the mRNA that also prevent from the degradation of the mRNAs. But again, these secondary structure formation also can either attenuate process of translation of the other genes, as well as it can also influence the transcription of the other genes, and we are going to take a look at those. And there are some small RNAs which bind to these mRNAs that can also control. So there are many other different types of control, and this just gives you an self-explanatory overview of the whole process. So let's take a look at the regulation of transcription initiation, which saves a considerable energy and materials. And we are going to take a look at this overview of this process. So the first thing that why we need to regulate that process, the first thing that is needed, that we have many genes in our body or in the bacteria as well, which we called as a constitutive genes. These constitutive genes are called the housekeeping genes that are expressed continuously in the cell. Say for an example, there are enzymes which are required for the DNA replication, or say from sodium potassium transport, or for the glucose uptake. These are many of these genes, thousands of genes. Those are needed by each and every cell all the time. And we call these genes as the constitutive genes. And these constitutive genes produce the mRNA by the transcription, and they produce by translation, the specific proteins that carry out the livelihood of these organisms. Now what happens that over the period of time, these particular proteins will get degraded. They are enzymes, they are working hard, and over the period of time, proteins degrade. They don't work forever. So you have to replenish them. So when their amount goes down, the cell exactly know when to replace them. So if the cells are constantly making all the proteins of the constitutive genes all the time, there may be a wastage. and the cell don't want the whole process of the transcription, translation. All these processes are extremely energy intensive. You need a large amount of ATP, and the cell want to conserve energy all the time. That's the goal, saving the energy. So all these constitutive genes, when they are needed, they are produced into the products. When they are not needed, they will be turned off so that the products are not formed unnecessarily and go into the waste. On the other hand, there is another group of proteins which work in the cell, which we call as an inducible genes. These inducible genes that code for inducible enzymes needed only in certain environment, such as beta-galactosidase. So again, we will go into the detail. For an example, of just giving an example, say for E. coli. So the E. coli don't have lactose in its natural environment, but it can grow in lactose. So what happens that if E. coli doesn't have lactose in its environment, all the proteins or enzymes that are needed for the metabolism of lactose are turned off. They are constitutively off. And what happens that they can be turned on, or they can be induced to be on when the lactose is present. The same way, there are some genes which can be turned off. They are usually all the time turned on, but they can be turned off when needed. A classic example we're going to see after that is a tryptophan. Tryptophan is an amino acid that is continuously required for the protein synthesis in all cells, including the E. coli. So this tryptophan is produced by a number of different enzymes required, at least five enzymes required in this process. And these enzymes are being made in the cell all the time. Now, if the tryptophan level is high, where there is no need for making more tryptophan, then the cell will turn it off. And we will use another word for this. These are inducible genes. That means that they are off, can be turned on. Whereas we are going to use another word which are called repressible genes. That means these repressible genes are turned on all the time and can be turned off. So inducible genes can be turned on, repressible genes can be turned off. So this is how the regulation of transcription can happen. So here is an example of these inducible genes, beta-galactosidase. Beta-galactosidase is an enzyme which is required for the lactose metabolism. This is a catabolic pathway. That means that we are using a catabolism. That means the bacteria is getting a sugar, which is lactose, that needed to be broken down into its monosaccharide, which is glucose and galactose. So inducible enzymes are present only when their substrate or the inducer effector molecule is available. So the inducer effector molecule is lactose here. And what happens? The beta-galactosidase catalyzes the hydrolysis of lactose into galactose and glucose. So this is the lactose and the beta-galactosidase. This is a side reaction which makes a product called allolactose right here. And that can be broken down into the glucose and galactose. These are the monosaccharides. So this is done by the beta-galactosidase. Now, the beta-galactosidase is not produced by the E. coli all the time. So these enzymes or the genes for these enzymes are turned off. Only when lactose is present in the environment, these genes are turned on. That means they are induced to make the beta-galactosidase protein, which is an enzyme, to do this reaction. Otherwise, they are not present. So just giving an example that if lactose is not present in the environment, usually a E. coli cell will have about 3 molecules of beta-galactosidase. On the other hand, if the E. coli is growing into a lactose media, each cell probably will have 3,000 molecules of beta-galactosidase enzymes. So this gives you an idea what an inducible or being turned on gene means. So on the other hand, we already talked about that. Repressible genes, the enzyme function that in biosynthetic pathways are product of repressible genes. Generally, these enzymes are always present. As I said, they are always on, and the end product in the biosynthetic pathway is available. So when these enzymes will be turned off, if the product is made in too much and in excess, so that there is no wastage. So let's take a look at the control of the transcription initiation by the regulatory proteins. So both induction and repression occur because of the activity of regulatory proteins and DNA binding domains. And we are going to take a look at these. These proteins inhibit the transcription. This is called a negative control, or they can promote the transcription. This is also known as a positive control. And there are much more complicated process in this whole process. So first, we are looking at the negative transcriptional control. So in the negative transcriptional control, the binding of regulatory protein at a DNA regulatory site, initiation of transcription is compromised. So here the mRNA expression is reduced. So again, if you recap from the previous chapter, that to make the mRNA from the DNA, the RNA polymerase, which transcribe, need to bind to a specific region of the DNA, which is known as the promoter. And near the promoter, there is also another region that we're going to see is called an operator. So this operator binds to different kind of things and help the binding of the RNA polymerase to the promoter. And these can help either the transcript to be made or not made. If RNA polymerase is not allowed to move through the promoter and operator into the DNA sequence or the template sequence, then no mRNA will be made. And this is the control of the transcription. So the repressor proteins are specific proteins that are present in an active and inactive forms. And these repressor proteins are inducers and co-repressors, which alter the activity by the repressor binding. And we are going to take a look at this whole process so that it makes a little bit easier. On the other hand, we are going to look at a positive transcriptional control. This is a binding of a regulatory protein at a regulatory region of DNA, promotes the transcription initiation. So the last one we talked about, that if the repressor binds, that stops the transcription. Transcriptional control protein binds, the mRNA synthesis is increased, and it can activate the protein activated by the inducer, and the active protein is inactivated by the inhibitor as well. So these are some of the levels at the transcriptional level. So this picture gives a very clear idea that how this transcription actually works. So let's take a look at that, what it means. That first, in that picture that we are seeing, that this is a negative control of an inducible gene. And if you carefully look at that this is, suppose this is the DNA, it doesn't matter whether 5 prime, 3 prime, just think about the DNA, and it is binding to the template region. And this is your promoter, and this is your operator. We talked about that. So you have a promoter and operator just upstream of your gene of interest that is needed to be producing your mRNA. So this repressor protein is binding to the operator region. So as this repressor protein is binding to the operator region, no RNA polymerase can bind into the promoter region. So what happens that, on the other hand, here we are looking at another type of regulation. Right here, there is an inducer molecule, and this inducer molecule can bind to the repressor protein. If the inducer molecule binds to the repressor protein, it no longer can bind to the operator and it will fall off. So here, without the inducer, the repressor bind to the operator. RNA polymerase cannot bind to the promoter. Here, the inducer is binding to the repressor molecule and blocks it from forming the structure, or it blocks formation of the interaction in between the operator and the repressor molecule. So at that point, the operator is empty. So as the operator is empty, the RNA polymerase can bind to this region, which is the promoter. So RNA polymerase can bind to the promoter region in absence of the repressor in the operator, and it can synthesize the mRNA. So this is one of the regulation that we actually can see. On the other hand, here we are looking at negative regulation. So this is the overall process, and we are just looking at a positive and negative regulation. What do we mean by that? So here we are looking at the same condition, but this is a negative regulation. So what we mean by the negative replication, right here, you have a repressor, and we call this repressor as an aporepressor. What we mean by the aporepressor, that this repressor do have a site for a co-repressor to bind. Until and unless that repressor binds to this repressor, it doesn't have the ability to bind to the operator. So just only the repressor is present. It cannot bind to the operator. RNA polymerase make the transcription. So there is no attenuation of transcription. The mRNAs are being made. Everything is good. On the other hand, what happened when this co-repressor is present, the co-repressor bind to the repressor, and now this repressor molecule is called an hollow repressor. So without the co-repressor, this is an aporepressor. When the co-repressor bind to the repressor, this is a hollow receptor. Hollow repressor bind to the operator region, doesn't allow the RNA polymerase to bind to the promoter again, and is the same condition like this that blocks the transcription. So this is the negative control of this process. On the other hand, there could be a positive control of the inducible gene as well. So here there are two different things. Here this gene is a repressible gene. That means that this gene is turned off when the repressor binds to the operator region. On the other hand, this is an inducible gene. What we mean by this inducible gene, that when an activator binds to the operator, that actually allows the recruitment of the RNA polymerase. So this is an activator protein. Without that, there could be some kind of transcription, but this really activates the transcription much. So this activator protein here is an apoactivator. So this apoactivator does have an empty site, which is an inducer that is not being bound. So the apoactivator cannot bind to the operator region. But here you can see that the inducer is binding to the activator protein. Now it is a hollow activator. It can bind to the operator region, and this actually helps in the recruitment of the RNA polymerase to the promoter. And again, just this is a cartoon. This really does not mean that, because you will have an impression that if this is binding to this, that will prevent the RNA polymerase to bind here. If this is binding in here, it will prevent the RNA polymerase from binding here. But this is just a cartoon. Two are entirely different things. Here, binding of the hollow repressor doesn't allow the RNA polymerase to bind, whereas think about that, this is like a clamp which actually tightly binds the RNA polymerase onto the DNA, which activates the transcription. On the other hand, here we are looking at a positive control of a repressible gene. So what we mean by a positive control of a repressible gene, here, just right here, this is the protein which is bound onto the operator, and when it is bound to the operator, it is actually making the mRNA. So there is no problem with that. So this is an activator protein, and the activator protein is bound to the here to a repressible gene, and it is turned on all the time. It's turned all the time. Don't think about this. So it's turned all the time. But what happens that when there is an inhibitor molecule right here, this is an inhibitor molecule, and when this inhibitor molecule binds into the activator protein, that activator protein will now fall off. And if this falls off, this one, which is a repressible genes, will actually turn off. So here is another control. Now remember that all these controls actually happen in different conditions at different times. So now we are looking at the decision process and the enzymes of the catabolic pathway only needed for the increased mRNA synthesis when preferred substrate is available. On the enzymes are not synthesized when the substrate is absent, and this is for the efficient use of energy and the material. So if we just look at the regulatory decision, let's see, this side is an catabolic enzyme. So this catabolic enzyme is the substrate of the pathway is present or not. This is the question that the organism is asking. If the substrate of the pathway is present, yes, then preferred carbon or energy source present. If it is yes, okay, all right, then we do not need that enzyme, so this is turned off. So do not synthesize enzymes. If the preferred source is not needed, no, then the synthesis happens and this particular gene is turned on. So this is an inducible pathway. These genes are usually off. They are usually off, can be turned off if the proper source is not available. Just the example of that, suppose we are right here and we are looking at a substrate for the pathway present, and we are looking at that we need to synthesize energy, and after that, okay, synthesizing energy, preferred carbon source and is present or not. Suppose when we are asking the question here, glucose is present. Okay, glucose is present, no need to do anything. That's perfectly fine. Nothing is needed. Here, we need to synthesize energy or make energy molecules, ATP, but we do not have glucose. Do we have lactose, something else? Yeah, we do have lactose. Okay, all right. And then turn the lactose genes on, we can synthesize lactose. So that's the overall idea. Here, on the other hand, are biosynthetic enzyme, or also, as we saw, say there's catabolic enzymes. We can think about that in this way, that these are anabolic enzymes. So end product of the pathway is actually present. So here, think about that. Tryptophan is an amino acid which is needed to synthesize proteins in the cell. And the question is being asked, is tryptophan present in the cell? Yes. Okay, all right. There is enough amount of tryptophan. There's no need to turn on the genes which make tryptophan. So turn them off. Then we are asking the question, is there enough tryptophan? No. Okay, all right. Turn on the genes to make more tryptophan because that is needed for the protein synthesis. This is the decision process in gene expression. So here we are looking at the negative control of the lactose operon and lac operon. And this is also from a little bit of prereq material, but just an overview of this process. I'm just, again, reminding that how this works. So here, what we have this lac operon. What it means by the lac operon, that these are some of the genes which are clustered together, which perform a specific function, which is utilization of the lactose. So what are the different parts of this lactose operon? So the first part, you have 3 structural genes. These are structural genes, although named structural, they really do not produce an equalized structure. They actually produce a protein, which are all enzymes to do a specific function. The first one, LAC-Z, it encodes the beta galactosidase, which make either allolactose or break down the allolactose or lactose into glucose and galactose. That's one of the main enzymes. Then the second one is right here, is a LAC-Y, encodes lactose permease. This lactose permease is a transporter which needed to be onto the plasma membrane to transport lactose from the environment. And here there is a galactose transacetylase, and the function of this is kind of a controversial, not really clearly known. But these are the main enzymes that is needed. Now they are under the control, and then of course you have a lac terminator at the end of these mRNAs. So think about that. These are all your polycistronic mRNAs can be produced out of that. Then upstream of these genes, you have the lac promoter where RNA polymerase binds, and then you have an operator which is bound to your repressor, inducer, all these other molecules, which helps the recruitment of RNA polymerase to the promoter. So you have an promoter and operator. These regions are in the upstream. Then you have another site which is called a CAP, or a catabolic activator protein binding site. So the CAP site is usually used as an activator when there is a particular catabolic activator protein is needed, and we're going to talk about that in a second. Upstream to that, there is another gene which is called LAC-I, and this is a regulatory gene, and it produces constantly a repressor molecule. Remember that the LAC-I produces a repressor molecule, and this is being produced all the time in the cell, which is under a LAC-I promoter. There is a separate promoter for the LAC-I, and constantly in a cell, in this whole section, LAC-I promoter binds to the RNA polymerase all the time, making the LAC-I product, which makes a repressor. So this LAC repressor binds to right here, lac repressor bind to the operator. Right here, bind to the operator. When this repressor binds to the operator, it will not allow the RNA polymerase to bind to the lac promoter. And that's the reason that lac Z, Y, or A, none of them are going to get transcribed in a normal condition. So now what will happen? We are taking a look at how lac repressor actually works. So here you have a lac Z and you have the separate operator sites. Here you have promoter, and the operator have an O1, O2, and O3, all these different sites. A typical structure, lac repressor, which is a dimer, that either makes an O1, O2 or O1, O3, but no matter what, when it binds to this region, it makes a bend in the DNA structure. It makes a bend in the DNA structure, and the DNA bends like this right here. And this will not allow the RNA polymerase to pass these structures, and that's the reason that the gene is constant in repression mode. What we mean by the repression mode, that there is no transcription and there is no product is formed in a normal condition. So on the other hand, what you are seeing that right here is a catabolic activator protein that is bound to the DNA as well. And the catabolic activator protein binds to this cap site right here. But even when the catabolic activator protein is bound in here, which helps in the enhancement of the transcription, if the repressor are bound in here, no matter what, even if the catabolic activator protein is bound in there, the RNA polymerase cannot go anywhere and cannot do anything. So really doesn't matter. If the repressor is bound, then that's it. That there is complete turn off of transcription. Now let's take a look at the regulation of the lac operon by the lac repressor and the whole overview of that. And we will also touch about how the regulation of this cap activator or the catalytic activator protein or the cap protein actually works in a fine regulation of this process. This is not too much discussed in your book, but we will cover that to make sure that we understand what's going on. So first, we are looking at the lac operon. And here the condition is that no lactose is available in the environment of E. coli. So what happens at this point? The LAC-I, this gene is under the control of the LAC-I promoter, separate. And as I said, that this constitutively, that means all the time, produce the repressor molecule, produce the mRNA, produce the repressor molecule by translation. And this repressor molecule binds to the operator region in between O1, O2 or O1, O3, and it blocks the RNA polymerase to move through the operator to produce LAC-Z, LAC-Y, or LAC-A mRNA. And there is no translation or there is no transcription, and due to that, there will be no translation, no production of any of the proteins that are needed for the utilization of lactose. So nothing is produced, and the repressor turns this whole operon off all the time. This is the constitutive condition. Now what happens that if there is a condition where E. coli is being placed into a media where you have lactose. So in this case, what happens? So when you place that E. coli into the lactose, a few molecules of lactose will come inside the cell. So your question will be how it is coming inside the cell. Now, just reminding you that these LAC-Z produce a beta-galactosidase and LAC-Y produce a permease, which is the lactose permease, which are inserted into the membrane so that the lactose can come into the cell. So in normal condition, when there is no lactose, sometimes, this is the important part, sometimes these lac repressor may fall off from these operator sequence just by chance. It's a binding process and it may fall off. When the repressor falls off, the RNA polymerase can synthesize just a few transcripts of the LAC-Z, Y, and A. They produce the mRNA and they will produce the proteins. So a few molecules of these proteins are available in the cell, even if lactose is not present. What is the difference? If lactose is not present, say, for example, there is about 3 molecules of beta-galactosidase present in a cell. Whereas if lactose is present, that molecule number can go up to 3,000. So just a small molecule. And we call that, in some other books somewhere you can find that, we call this as a leaky expression. Think about that. You have a faucet where you close the tap and your intention is to turn off the water, but the valve is not proper or worn out, and it will drop the water drop by drop just a little bit. And that is the exact same thing what we mean by a leaky expression of these LAC-ZY and A protein can happen in absence of lactose. So when the E. coli is present in a media where you have lactose, the amount of the leaky expressed proteins, which is the permease, will bring a few molecules of lactose inside the cell. And those few molecules of lactose coming into the cell will be converted into allolactose by the beta-galactosidase. Again, just a few molecules of beta-galactosidase that is present in the cell. Once those are converted into allolactose, they will start binding to the repressor. And the repressor have the binding site for this allolactose, and allolactose is an inducer. So the inducer will bind to the repressor and it will make the repressor inactive. If the allolactose, which is an inducer, bind to the repressor, it will not be able to bind to the operator anymore. And this is the inactivation of the repressor by the allolactose. So if they cannot bind to the operator anymore, RNA polymerase will be recruited to the promoter, and all these genes will be transcribed into the mRNA and proteins will be produced. So this is the condition that is happening in presence of lactose. As more and more permease is going to be produced and going into the plasma membrane, more lactose will come in, more beta-galactosidase is being produced, so more allolactose is going to be produced, and all the repressor eventually will be completely blocked. So there will be a high level of transcription of the mRNA for all these LAC-ZYA genes, and lactose will be utilized for the purpose of metabolism. Now, where this cap regulation comes in. So this is an interesting part, that the cap binding protein binds to a site which is just here, right here, which was mentioned in a previous slide, I guess here, the cap site right here, the purple one. So what is the function of this cap binding protein and what it actually does? Now, let's very carefully listen to this. So if there is a condition when there is glucose in the environment as well as lactose in the environment, again, glucose in the environment as well as lactose in the environment, the E. coli prefers to allow preferential use of glucose. So it will utilize the glucose and it will utilize the lactose, but not that much. So what is happening here? So let me tell the other scenario. If there is a condition when there is no glucose in the environment and there is only lactose in the environment, at that time there is a particular molecule whose level goes up in the cell. That is cyclic AMP or CAMP. This cyclic AMP or CAMP bind to the catabolic activator protein, and when the CAMP bind to the catabolic activator protein, this binds to the cap site. This, again, CAMP level rises when there is no glucose. That binds to the cap binding protein, that will bind to the cap site. When the cap binding protein binds to the cap site, it recruits the RNA polymerase much more tightly, and it will make a high level of transcript in that condition. When your glucose level is high along with the lactose, what happens? The CAMP level is low. No cap binding protein will bind to this. RNA polymerase will transcribe these lactose utilization genes, but not that efficiently because sometimes it may fall off. If cap binding protein is in there, it makes sure that it binds the genes much more tightly through the promoter and the rest of the way, so that the amount of transcript made is much more. and this allows the preferential use of glucose. I'm just taking an arbitrary number that may make it easier. If glucose is present in the lactose, the binding of the RNA polymerase and making of these genes may say that the efficiency of the RNA polymerase is at that time about 60%. So 60% of the time, the RNA polymerase will make these transcript and make these lactose utilization gene when glucose level is high. If the glucose level is low, that means the cell understands there is no other energy source. Your only source is lactose, which you have to break down into galactose and glucose to get the energy. Then the cAMP level rises, the cap binding protein tightly attaches the RNA polymerase to the cap site, and now the efficiency of RNA polymerase to produce these transcript goes from say 60% to 95%. So this is the advantage of glucose being present and not present along with the lactose. So this is the overview of that when lactose is not present, there is no production of any inducer, so that all the repressors stop this production. Lactose present in presence of glucose, the efficiency of the production of the lactose utilizing genes and the production of the mRNAs is a little bit less because there is no cap binding protein. When there is no glucose and only lactose, cAMP is produced in the presence of low glucose that will bind the cap activator protein, and that will allow the highest efficiency to produce all the transcripts at the maximal level to utilize all the lactose available for the functioning of the operon. So this is the fine control of the lac repressor or the lac operon. So the next one we are going to talk about is the opposite, which is the tryptophan or the trip operon, which consists of 5 structural genes. So here we are looking at the promoter in green. Then we have this region, this is the trip R, is making an inactive repressor. And this repressor in inactive, that's an aporepressor. So it's inactive. This is your promoter region, operator G region. The structure of this operon is a little bit different. So the first region is called the trip L, and the trip L is actually a leader sequence. There is a leader sequence. Then you have an attenuator sequence in the red. So this is a leader and attenuator sequence. Then you have a trip E and trip D. So these make some specific proteins, which is called anthranilate synthase, anthranilate, and this anthranilate synthase is a pathway enzyme. Again, you do not need that for tryptophan. Then you have an anthranilate isomerase that is made by the TRPC, and then you have 2 types of tryptophan synthase beta and tryptophan synthase alpha. So this anthranilate synthase, anthranilate isomerase, and tryptophan synthase, these are the enzymes of the metabolic pathway of tryptophan, are made by this trip operon. In the normal condition, RNA polymerase synthesizes all these enzymes, and tryptophan is needed in the cell for all the protein synthesis that is going on. So this gene is turned on all the time, making an RNA by the transcription and translation. And the transcription and translation is also coupled. If the level of tryptophan in the cell goes up, tryptophan by itself is called a co-repressor. Why it's a co-repressor? Because you have a tryptophan repressor molecule. It is inactivated by itself, so it's an aporepressor. Tryptophan by itself can bind to the co-repressor. Now it becomes a holorepressor, and this holorepressor binds to the operator, and now there is no more transcription. Transcription is off because there is enough amount of tryptophan in the cell. So that's shutting down the tryptophan synthesis. So that's the crude way of turning off the synthesis of tryptophan. But there is a little bit finer control compared to this just turned on and turned off. Because here what we are seeing that when there is a high tryptophan level, repression occurs and there is no translation of tryptophan happens either because there is no mRNA. But what happens at one point of time, tryptophan is going to get depleted. There is not enough tryptophan. There's a low amount of tryptophan, at that time, you need to start transcribing tryptophan and you need to translate tryptophan as well. And then again, when you have high amount, you turn it off again. And we have to take a look at these controls in a little bit detailed way. So before we go into the trip operon into the detailed way, which we called as an anti-termination or attenuation, before that, let's take a look at another operon, which is an arabinose operon or ARAB. So here, the arabinose by itself acts as a both positive and negative regulator. So what you are seeing in this picture, the same way you have these ARA B, A, and D. These are all your the structural genes. You have a catabolic activator site, you have an operator and a promoter. So here you have an arabinose operator and you have an ARA O2, O1, and you have an L. So you have ARA C protein, which forms a dimer arabinose binding domain right here. Along with that, these dimer region forms a dimer in between ARA A2 and ARA L. When this dimerization happens, the DNA bends, the RNA polymerase cannot go through this, and there is no transcription. So this operon is inhibited in absence of arabinose. But there is also this ARA C protein binds to that ARA O1 region as well. So here you have two molecules of ARA C, here you have one molecule of ARA C, one molecule of ARA C. So there's altogether 4 molecules of ARA C, so no transcription. Now what happens when you have enough amount of arabinose, there is a particular area which is the arabinose binding domain, just pointed right here. This whole is the arabinose binding domain. When arabinose is enough in the cell, arabinose will bind into these pockets. So when this happens, and you can easily see the loop in between right here, the loop in between this ARA 2 right here and this ARA L is broken. On the other hand, you can see that right here, there is a dimerization of 2 proteins right here at the ARA L and ARA O1, you already have 2 proteins right here, and they all recruited the arabinose. And at that time, the arabinose bound to this ARA C protein will work with this CAP protein. If you remember that, the catabolic activator protein, we talked about that the cyclic AMP or CAMP is needed for the catabolic activator protein. And this will recruit the catabolic activator protein bound to CAMP to the CAP site. And this is where the arabinose operon will be activated in the presence of arabinose. When there is no arabinose, there is no need for these genes because you do not need any of these genes to metabolize arabinose because it's not present. When arabinose is present, they are binding to form this loop so that the RNA polymerase can be recruited properly to the promoter region, and again the CAP site enhances the recruitment of the RNA polymerase to synthesize all the genes needed for the utilization of arabinose, which is a sugar. And this is an important sugar for the pentose phosphate pathway to make nucleotides. So the next part we are looking at the attenuation and the ribose switches, which can stop the transcription prematurely. And we are looking at this particular trip operon in a much more detailed way. So here, the regulation of the transcriptional elongation. Transcription can also be regulated by controlling the transcription termination. Not only initiation, so far we saw the initiation, but it can be controlled by the transcription termination as well. And this type of regulation is called an attenuation of transcription. This was first demonstrated with the trip operon, and more recently, the ribose switches have been demonstrated also to play a regulatory role as well. So if we are looking at the trip operon attenuation, the transcription continuation is also controlled by this operon. And this attenuation termination is within the leader region or the leader peptide and occurs through the stem lip structure in the mRNA, depending on the level of the tryptophan. So how this actually works. So right here, what you are looking at, this is the leader sequence from 5' to 3'. let's take a look at where is this in the trip operon. So if we are looking at the trip operon right here, this trip L, this purple sequence right here, this is the leader sequence with the attenuator sequence red after this. So we are looking at just only that region. So that region is this. So here we have some interesting sequences. We have a region 1, 2, 3, 4, and then 3' end. In the region 1 can base pair with region 2 because they have palindromic sequences. So if you have a sequence this way, you have a sequence backward this way. So if you have a palindrome which reads forward like this and backward like this as well, then they will all base pair. So 1 and 2 can base pair. Interestingly, the region 2 and 3 also have palindromic sequences. So 2, 3 can base pair as well. And 3 and 4 also have palindromic sequences. So 3, 4 can base pair. So no matter what, 1, 2 can form stem loop, 2, 3 can make stem loop, 3, 4 can make stem loop as well. So in addition to that, the region 1 of this leader sequence has these 2 red lines or slash right here. There are 2 tryptophan sites. These are 2 codons of tryptophan, so that these are built into the peptide. So this mRNA is going to make a leader peptide, and that leader peptide does have these 2 tryptophan in there in the region 1. So what happens that when there is no translation occurring, means that at one point of time, when there is enough amount of tryptophan in the cell stopped transcription, there is no translation occurring in the cell of tryptophan. At that time, region 1 and 2 and 3 and 4 base pairs, and this helps the RNA polymerase to fall off, because RNA polymerase cannot go, if this loop is formed, RNA polymerase, this is following that, you have an U-rich sequence. Right here, you have a lot of U-rich sequence. So this is attenuator, and when you have these stem loops just prior to this U, RNA polymerase will fall off. There will be no transcription further. Now, if there is no transcription further, remember that your tryptophan E, D, B, A, they are all downstream, and the RNA polymerase, if falling off from here, it will not be able to go. So there will be complete transcription attenuation. Now, if you remember that, we talked about the row-independent transcription termination, if you remember from the previous chapter, which forms a stem loop, and next to the stem loop, there is an U-rich region, and the RNA polymerase falls off. This termination is the same way, which is the row-independent or row factor independent transcription termination, where you have a stem loop structure followed by a U residue where RNA polymerase falls off. So this is the type of attenuation that will happen when there is no translation occurring. And why there is no translation occurring? The reason for that is probably you have high tryptophan level. There is no more tryptophan needed. So no transcription. Now what happens that this is a subtle difference. At this point, the translation is occurring. Why it is so? Because at this point, there is low tryptophan level. So the tryptophan is abundant. There's tryptophans being used up. Now you have a low tryptophan, and those low tryptophan need to start translating. So let's see what happens when you have a low tryptophan level. So here, the RNA polymerase is transcribing these, and the ribosome is following the RNA polymerase to translate. Now, this region 1-2 loop cannot form. What is the problem here? See what is happening, that in the region 1, there are 2 tryptophan sequences. As the tryptophan, this is the important part, as tryptophan level is very low, it may take some time to load the tryptophan onto a tRNA, and this amino acid tRNA site is devoid of a tryptophan for a while. So here in the tryptophan, when the mRNA comes to this tryptophan codon, the ribosome stalls at this point a little bit. This ribosome stops right here. because it takes a little bit of time for the second tryptophan to come in, which is at the second time. So ribosome stalls. So as ribosome stalls right here, it is taking this region, a portion of the region 1. So your loop 1, 2 cannot form. On the other hand, instead of that, what you are seeing that as it is stalling on the region 1, you have a region 2, 3, which is region 2, 3 is base pairing right here. This is called an anti-terminator. What do you mean by this? That if the region 2 and 3 base pairs, RNA polymerase will not stop. RNA polymerase will only stop if you have a 3-4 loop next to the U region. 3-4 is not making a loop. 2-3 is making a loop, which is away from the U residue. The RNA polymerase goes on, transcribes, so the transcription doesn't stop. So 2-3 is called an anti-terminator, no termination of transcription. And this is done by the pause of the ribosome at the number 1 codon of the tryptophan right here, while it's waiting for the second one to come in. That pause causes the engagement of region 1, and there is no region 1, 2 loop. And as the 2 is empty, the 2 and 3 immediately forms the loop. Now, the third case, now you have translation is occurring and you have a high tryptophan level. In the high tryptophan level, what happens that as you have enough amount of tryptophan, the ribosome goes past the number 1 region very fast, and it goes to the number 2 region quickly as well, because it will not stall here. Here it was waiting for another tryptophan because tryptophan level was low. That's why it's pausing. Here tryptophan level is very high, so there is no problem for finding a tryptophan by amino acid by the tRNA. So tRNA will recruit here the next tryptophan immediately. So this will go with the region 1 and 2 quickly. As it is going through the region 1 and 2, so the 2 is engaged as well. So 2 cannot form a base pair with 3. The unengaged areas are 3 and 4. So 3 and 4 will form a stem loop here. As 3 and 4 forms a stem loop, next to that is a U-rich sequence. Again, RNA polymerase will fall off. Now here you are seeing a high tryptophan level. You need to turn off the transcription. And this is the, again, is the transcription terminator loop, turning off the transcription. So this is at the very beginning when there is no translation occurring because you have already high amount of tryptophan in the cell. When it is depleted, level low, it is anti-termination happening and it is allowing the transcription to happen. And when there is enough amount of tryptophan level reached into the cell, it will stop the transcription as well. So this is in addition to the control where when tryptophan is already made enough amount, remember that tryptophan when made in good amount, tryptophan just by itself will start even the initiation. So this control is at the initiation level because tryptophan is binding to the repressor and it will not allow the RNA polymerase to make even the leader. So this is a one level of transcriptional control at the initiation, whereas this control is at the termination level. So this is a second level and it's a much more finer control of the transcription. So that's pretty much all of it. And you will see that there is another example of the ribose switches, which just gives you a picture in your book, which shows that how these ribose switches can sense the environment, and they can also form these loop-like structure where the formation of the material, if it is being produced, will bind to one of these structures and disrupt that so that either the product will be made or it will not be made. So if the product is not available, it will be made. If the product is available, it will turn off this product by these ribose switches, and you can take a look at those. And that will be pretty much the overview of this process, that how the bacterial processes of gene expression is controlled at the transcription and translational level, at the transcription initiation level, as well as the transcription termination level as well.